Olá a todos, boa tarde. Eu gostaria de agradecer a apresentação de todos que se inscreveram no webinar da Cellenion, para conhecer um pouco mais das soluções, né, da Cellenion, sobre as aplicações com single cell, com tecnologia para soluções para cultivos celulares 3D. E hoje a Uniscience tem o prazer de trazer aqui o especialista de single cell e células 3D da Cellenion, né, o Tylor Rockwood. O Tylor ele possui bacharelado e mestrado em bioquímica, com foco em química medicinal pela Arizona State University. É especialista de aplicações hoje na Cellenion e vai falar um pouco sobre as soluções para single cell e modelos celulares 3D. Então, so Tylor, thank you very much to stay here with us today. And when you are ready, please, you can start the presentation. Thank you.
Thank you for the introduction. So hello, everybody. I'm Tylor Rockwood. As you said, I'm an application scientist with Cellenion, and I'm here today to talk to everyone about the CellenONE and the SpheroONE, which is our answers to single particle isolation, both cells up through three-dimensional models. So we have the 3D model you need, and let's get started. Now, first, I want to mention that Cellenion and Cellenion are not standalone companies necessarily. We are part of a larger ecosystem of technology organizations represented by BICO. And so this is not something that we are a small entity. We have a lot of support, and we also have a lot of connections that create a much larger capability when it comes to technology and instrumentation. So primarily, I'm going to talk to you about our CellenONE line, which is our flagship model. This covers particle sizes or cells that are from a 0.5 micron up through an 80 micron range, and does gentle isolation of these as well as some other functions on system, which we'll get into here. Now, the SpheroONE is our newer system and is currently available as a custom solution. This expands that size range to where we're targeting spheroids, organoids, and tumoroids between a 50 micron up through a 600 micron range. So we're going to start with the CellenONE, the all-in-one single-cell platform pictured here. I want to show that this is a freestanding platform, so it does not require any bench space, and it is self-contained. Now, this platform is capable of performing single-cell isolation, but it also performs precision liquid dispensing and on-deck incubation, which reduces the number of hands-on steps needed for the processing of samples rather than just being used to actually isolate the cells. This is our flagship product. It is the most precise system, combining both the single-cell isolation and nanoliter dispensing in one device. Apologies, it's delayed a bit here, moving to the next slide. Can you still hear me? Seems my screen is frozen here. Hello?
Hello, Taylor. Can you hear me?
I can hear you now. Apologies,
Great.
I've been frozen on one screen for a bit.
Yes, we can see. We are in the slide cell 1, and when you put 1, 2, and 3, right?
Yes. I'm attempting to move to the next slide, but it seems to be hesitant. I'm glad you can hear me again, though. So while this waits, as long as you can hear me, I'll talk a little bit about this, which is in order to perform these three functions, the system uses acoustic-based precision dispensing. I'm still attempting here to move on to that next slide. But this means that it is a gentler sorting method. It also uses image-based cell isolation, and the benefit of that is that it allows us to take an image of each individual cell before isolating, and we can then track that along with the metrics that are measured on it, so that every single well, every single cell isolated, we have a record of exactly what it was. I seem to be I seem to be fully frozen again. Can you still hear me?
Yes, we can hear you. Now we are seeing the presentation again.
Okay.
Great.
All right. I seem to be
Yes,
able to move
great.
forward. I can move backwards, but not to the next slide.
Oh,
So
okay.
I'm going to. Okay. We are moving forward again. Apologies for the delay there.
Okay, great.
All right. So the platform does an acoustic-based precision dispensing. This allows us to get a very, very precise volume in the picoscale range, and then that allows us to build up and do very precise nanoliter scale reagent dispensing. We also use it for an image-based cell isolation, which means that we do take an image of every single cell that's isolated. We're able to measure some metrics on it, like the size, the elongation, the intensity, and we can measure fluorescence channels on that. And those are all saved and tied directly to each well or location that a cell is dispensed so that you can look back to it later. Finally, we have environmental controls on the deck, which allows us to do incubations and longer processes without having to remove the samples from the instrument. And those three together allow us to automate this workflow so that there's fewer manual handling steps. So I'm going to show a video here quickly, which just demonstrates what or how we actually perform this cell isolation using the CellenONE. Now, it is a modular system that allows us to customize the deck. We can have a variety of different plate types, including standard ones like a 96 or a 384 well plate. We also can do custom targets and have some proteo chips that we use in order to have a off-the-shelf answer to proteomics. The cells are aspirated into a glass capillary, and then once they are in there, we can image them as we do drop by drop and actually do isolation based on the image of each individual cell. An ejection zone is established where we know that the very next droplet will dispense anything that's within that zone. And then there is a sedimentation zone, which is a safety factor to ensure nothing sediments down into that ejection zone. Now, once a cell is detected and it is just a single cell in there, the glass capillary is moved over a target well, and that cell is dispensed and returns to look for the next cell. Now, each image is being analyzed, and so when a particle is detected, if there are multiple particles, it will not dispense, and it'll send that down into a collection vial. So if this is a particularly valuable sample or something that you don't have a lot of, you can take and reuse those cells. Each cell is also evaluated for a diameter, a circularity, elongation factor, and that is partially used to determine if it is a single cell or if it is some other particle, or for in this example, a duplex of two cells over each other, which will have a less circular appearance. Now, those are all metrics that you can use on the system, and you can determine where those thresholds are. You can also run an analysis of your sample directly there in front of the camera, which will give you a readout showing exactly what you have and allow you to choose the parameters you want to use for isolation. It can isolate any type of cells, provided that they are within the 0.5 to 80 micron diameter range. We can use bright field imaging for size and elongation, as well as 4 different fluorescence channels. And each of these can be gated to either require or remove a fluorescent appearance for the cells, meaning you can sort the ones that either do or do not have this fluorescence, and you can set the threshold of exactly where you want that line to be drawn. And this allows you to do very precise gating of exactly what you want isolated versus what you want to be passed over. The platform does allow for a very wide range of applications. Many of the ones that we currently work with are related to single cell omics. because of its capacity to find and isolate one specific cell into any number of different locations. You can also use it to isolate a specific number of cells by having it return to the same place with multiple cells. It does not need to be just one cell to one location. And this allows us to do things like spatial proteomics, single cell proteomics, single cell RNA sequencing, and so on. It can also be used to isolate cells that are remaining viable in order to be used for downstream processes as well. It does not need to be used for cells that are to be digested. I'm trying once again here to move to the next slide. Here we are. Okay, so I want to talk a little bit about single cell proteomics and the reason behind it here. So when you are looking at a tumor or a developing tissue or an immune response, it is a mixture of a number of different cells that are all at different types, different states, and they're all pooled together. The benefit of using this single cell isolation is that you can do single cell proteomics and resolve that heterogeneity and identify exactly what is happening in each of the different cell types or cell states through isolating documentation and then processing downstream. A lot of the information that you gain from this is the exact information that is lost when you analyze the cells in bulk. Now, unlike nucleic acids, proteins can't be amplified, which means that when you're doing different steps in your processing, every single manual pipetting, every single step can have a loss of your sample, and that can cause difficulty with analyzing the results when you get a lower amount of data out of your experiment. With single cells, this is really a great example because there is already very little material there that you're working with, and any small loss during sample preparation can drastically affect your results. So with the CellenONE, this is a complete open, it's a sample prep platform for single cell proteomics. And what that means is that we have the capability to use off-the-shelf methods, but we also have the capability of doing custom methods where it can fit the experiment that you need. Now, single cell isolation is done on the system. It also can use the system to do the reagent dispensing, the incubations and lysis, and then further than that, the inactivations and the dilutions, so that by the time your sample is ready to leave the instrument, it is ready to move directly on to the LC and mass spectrometry. The products you see here are our current set of Proteo chips. So these are off-the-shelf single-cell proteomics workflows that we offer. We do receive up to 5,000 proteins per cell on the latest mass spectrometry systems. For the label-free, there are 2 different label-free methods and 1 multiplexed method. All of these are capable of being purchased to where it can be directly loaded onto the deck. They are already validated, and a customer can directly move into using these after the purchase of an instrument. Now, this is just a composite figure from a paper from the Broad Institute, the CAR lab. And this is where, apologies here, THP1 cells, which is a monocyte model, were stimulated with liposaccharides versus DMSO control. And the entire sample prep was on a CellenONE with the ProteoChip Evo 96 with a direct transfer to Evo tips by centrifugation. That means there were no manual handling steps required for the cells in this process. And the bar chart on the left shows you something crucial because it is the direct transfer to Evo step using centrifugation versus a manual pipetting step at the very end to transfer over with the nano-elute. And the difference is twofold. You see that a single manual pipetting step at this scale can cause a 50% reduction in the number of protein groups identified. Now, This next one is a custom protocol using 1536 well plates. And this is just to demonstrate that although we do offer off-the-shelf protocols, that is not required that you use these. The system is capable of doing custom targets, custom plates, and can still be used to incubate and perform all of the steps on system. In this example, this is showing the optimization using a 1536 well format, where in the end it was found that 100 nanoliter volumes not only performed best in this 1536 well format, but actually performed better than the same volume on a 384 well plate. This is what we mean by this being an open platform. Now, this is just an example of some of the toughest critics in the world that have made the CellenONE their standard and are using it currently for proteomics. So from here, I want to move on to the SpheroONE, which, as I said before, this is a newer platform currently available as a custom solution, and this expands the size range of particles that we are working with up to encompass spheroids, organoids, up through 600 microns. Now, a primary reason for this would be with new approach methodologies in order to eliminate the need for animal models. There's many reasons to do this. The primary ones that we're showing is that it is poor translation to humans, reproducibility difficulties, experimental biases, economic inefficiency, ethical questions. With the SpheroONE, we are able to do automated isolation of spheroids. This allows us to avoid manual transfer steps. Because 3D models are very delicate, especially when it comes to, for example, spheroids, they can be damaged or destroyed in manual pipetting steps. Using this for isolation allows us to isolate while maintaining their integrity. It also allows us to gate these and use very specific sizes rather than looking at large populations. Show a little video of the SpheroONE here. There are some core differences with SpheroONE versus our CellenONE. Now, one difference is that this no longer uses an aspiration step. The sample holder here will be a tube and does an automatic resuspension of your sample spheroids. This also means that the priming of the system and loading of samples is more automated. This allows it to be a much more user-friendly experience, but in the end, we are still using image-based isolation, and we still have the 4 fluorescent channels that are used for isolating. And what we're going to see here is just a real-time isolation happening on our SpheroONE. Now you can see that we are also no longer using the same piezo dispense capillaries. We are using flat nano dispense capillaries for the SpheroONE. This allows us to get an even higher image quality here and will allow us to distribute as much as a 384 well plate in an average of 30 minutes with above a 90% single isolation range. The software has also been built once again from the ground up. This means that it is a very user-friendly experience in selecting your parameters for isolating exactly what you want to be isolated versus ignored in the system. It shows you at the same time a scatter plot, which includes information on each of the spheroids that have been isolated, images in the bright field as well as the fluorescent channels, and a running total of which positions on your plate have currently been isolated. And this is just to show that you can choose either a broad range when performing an isolation, showing the image on the left, where any different size of spheroids within this sample population will be isolated, but you can also pick a very narrow range. For example, the upper right image showing larger spheroids being isolated versus the lower right showing smaller. You can choose where those gates are, and you can also run an analysis of your sample prior to making this choice so that you can see what your population includes and then make your choice from there. This system does still have the same controlled target plate temperature, which means we can perform incubations as well and keep everything on system. Now, this study is one that was done in conjunction with Sartorius, and you can see a difference right here between, on the right side, manual pipetting of spheroids versus those that are isolated using the SpheroONE, and this is just from the gentle dispense capability. What was used is a drug treatment. Apologies. Effectively, they were testing a drug treatment for, I can't say this properly, camptothecin. And what they wanted to see was, with a very specific size range of particles, how much over time, using different concentrations of the drug, did it actually slow the growth. And what you see on the next slide here is that there's a very, very clear reproducible drug response. And this is just due to the ability to very specifically isolate spheroids, ensure that there is only one particle in each well, and monitor them over time, knowing that the viability is kept over 90% when isolating. Now, this is just showing two examples of a large gate or large isolation gate where there was a very wide set of parameters used for isolating and a very large difference in size and elongation of the different spheroids that were isolated versus a very narrow isolation range. And what we see is that when using a drug response assay with these, there are much lower error bars with each of the samples where there was a very narrow gate shown, meaning we can reduce variability in the testing and make it more viable to use drug response assays with spheroids versus the animal models. Okay. So the take-home message with SpheroONE here is that sorting and isolation of spheroids of interest for highly homogeneous populations, which is perfect for drug screening. It is gentle dispensing. There's lower waste, but it also reduces damage to the spheroids and organoids that are being isolated. Imaging is easy and allows for precise positioning. This allows you to do the reagent dispensing and ensure that you are dispensing your spheroids directly into the reagents. It allows for a much lower volume use as well versus traditional methods, and a high throughput, meaning up to 2 384 well plates in 45 minutes, and with a single isolation range over 90%. So recap here, the entire portfolio looking at the CellenONE line with the CellenONE Neo as our current model, and this is our flagship model. The SpheroONE line, which is our newest model and is available as a custom solution, and that collectively these represent the ability to very gently and accurately isolate particles from 0.5 microns all the way through 600 microns, while also doing reagent dispensing and incubation on deck and reducing manual handling steps. All right, thank you. You still hear me?
Yes, can you hear me? I'm
yes,
here.
yes. Apologies, I tried to speed up a little bit because I know I've been having the technical difficulties setting us back there.
So it was a really great presentation. Thank you very much. Very interesting, very relevant. We have a question from Pedro here. We are going to put the question in the screen. Can you see it?
I actually cannot see the question without removing the presentation. I'm on only one screen.
Okay, I can read for you. Pedro asked, how can I proceed to perform a cell suspension blank analysis with no cells with the CellenONE?
Okay. So what I would recommend for the blank analysis would be aspirating some of your media, whatever it is that you're suspending your cells in, and then you can tell the system, just like you can do reagent dispensing, you can have the system drop a single droplet of that cell media into the locations that you want to be a blank. So effectively, a negative control can be one where you just choose not to isolate a cell into it, but if you want to be even more accurate, you can intentionally dispense a single droplet of your solution, but that does not contain a cell.
Okay, Tyler, thank you very much. We have no more questions. Thank you, Pedro, for asking this question.
I do have, sorry, to follow up on that, I realized there is another method that I forgot to mention. With the CellenONE, with all of the parameters, you can choose to use a positive or a negative or a neutral, meaning that you can also just tell the system that you only want to isolate if there is not a particle visible. And that way you can use the actual suspension that has the cells in it, but without isolating a cell into some locations. The same goes for all of the fluorescence channels. You can choose to gate for cells that have above a certain threshold of fluorescence, below a certain threshold, or you can also just have it monitor the fluorescence levels while not using that as a metric for isolating cells. And those will all be saved, which means you can go back later after you have your results and say, okay, is there any correlation between those that were fluorescent and those that were not? Or what is the correlation?
Okay, thank you, Tyler. We have another question here in the chat. Alexandre asked, could you do a brief review on how CellenONE deals with cell heterogeneity?
So the primary focus with the CellenONE is the ability to analyze your cell sample, meaning it will dispense until it has detected 100 particles, and it will plot out exactly what the size and the elongation and the fluorescence levels and whatever you're looking at for those. And then it will allow you to use that to gate exactly how tight or how loose you want to use your isolation parameters. So when it comes to trying to make a very heterogeneous set of cells isolated, you can make very narrow gates where it will only be isolated if it's within these very specific parameters. Or if you want, or I'm sorry, heterogeneity, I'm going the opposite direction. If you want heterogeneity, then you can set very wide parameters, and you can make it so that it will isolate regardless of size or within a much wider range, but it will still document exactly what the parameters that are measured for each of the different cells isolated are. And it will also document where each of those go so that you can correlate exactly the metrics of one cell to where it ended up and then further down the line to your results.
Great, Tyler. Thank you very much. We have no questions more in the chat. I would like to thank you for this very nice presentation. It is very interesting. And I will just speak here in Portuguese a little bit for the people that are watching. Obrigada, pessoal, a todos vocês que assistiram hoje a nossa palestra com o Tyler. Se vocês tiverem outras perguntas, curiosidades, quiserem saber um pouco mais sobre as soluções da CellenONE, entre em contato com a gente da Uniscience, que a gente tem todo um time de suporte técnico, de assessoria científica, para auxiliar vocês em dúvidas, questões, para entender quais as soluções são mais adequadas para suas aplicações, para os seus laboratórios. Então, fiquem à vontade, se tiverem mais alguma dúvida, entrem em contato com a gente e a gente pode seguir conversando aqui também depois da palestra, tá bom, pessoal? Muito obrigada, um abraço a todos. Tchau, tchau.